Another class of lipids are the phospholipids, which are probably the major component of most of our membranes. Depending upon cells, that can vary, but in general, the bulk of our cells use phospholipids as their primary lipid in the membrane. And the reason for this is that basically they contain a molecule of glycerol here, and to that we have esterified two fatty acids, but we also have a molecule of phosphoric acid, or a phosphate group, attached as well. And as a result, we have a very hydrophobic region here in the molecule and a hydrophilic region that can interact with the aqueous environment that surrounds the cell. It just so happens that the space occupied by the two phospholipid tails here is about the same space that's occupied by the polar head group, so that you end up with a very cylinder-like structure for a phospholipid membrane. In addition to being found in our membranes, we also use these as emulsification agents. If you ever read ingredients on the food you eat, you might find the term lecithin, particularly in milk chocolate. This is an emulsification agent that is used in basically in making or allowing milk and chocolate to be mixed together. So the structures that are formed by phospholipids are largely referred to as a bilayer. And if we look at a typical phospholipid, here would be the phosphate head group, and then the hydrocarbon tails would be here. They generally organize in such a manner that the phosphate head group associates with the aqueous environment, which would be on both sides of the membrane. And in the middle, we have this very large hydrophobic region. And this actually gives rise to some of the properties of membranes. They self-assemble, getting the hydrophobic tails of the fatty acids away from the aqueous environment, allowing the phosphate portion to associate with the aqueous environment. Now, phospholipids very often have other items attached to the phosphate. And in general, if this is the structure of a phospholipid, the phosphate very often has something else attached to it. And the something else can be, for example, a molecule of choline. This is actually what lecithin is. It's called phosphatidylcholine. Choline basically is essentially an amino alcohol that contains a positive charge on the amine group. Ethanolamine is another amino alcohol that can be added. Inositol is a derivative, is a carbohydrate derivative that very often ends up on membrane structures as well. Other things that can be added would be serine to give phosphatidylserine, and then sometimes you can actually even add another molecule of glycerol in there, to which you can attach other molecules. So there are a large number of differences in the different types of phospholipids that we see present in our membranes. Now, another class of membrane lipids are the sphingolipids. And sphingolipids are much different than the phospholipids that we saw earlier. Phospholipids are derived from glycerol, whereas sphingolipids are derived from this molecule sphingosine. And sphingosine is basically an amino alcohol. And basically the amine group here and the alcohol group here are very often attached to other molecules. The amine group is actually attached to a fatty acid through an amide bond, as we see here, to give what we call a ceramide. In general, the fatty acid that's attached is a 14-carbon fatty acid. To the alcohol, very often we have attached a phosphate. If that phosphate also contains a molecule of choline, we get a molecule called sphingomyelin. Sphingomyelin is a component of membranes. It's found in very high concentration in the myelin sheath of nerve cells, where it derives its name. In addition, instead of the choline that we see there, very often at this oxygen here, we can attach carbohydrates, which give rise to molecules such as cerebrosides and gangliosides, which are other types of phospholipids or membrane lipids that are particularly found in varieties of nerve cells. Isoprenoids are completely different in that all the other lipids that we just looked at were derived from fatty acids, whereas isoprenoids are actually derived from the molecule isoprene, which we see here. From isoprene, we can build molecules called terpenes, which have their own uses in our cells. And terpenes are the basis for the generation of the molecules like cholesterol, which are classified as steroids. Isoprene itself is not a lipid that we very often would see present in a living organism. Instead, isoprene molecules get attached together such that generally we look for isoprene units in the molecules that are built from isoprene. So some examples of some of the simple terpenes that we see, basically this molecule geranyl is what we call a monoterpene. It's constructed from two isoprenes, so one of the isoprenes would be right here, the second would be right there. So those two isoprenes are brought together and undergo a reaction and ultimately form this molecule geranyl. We can bring in a third molecule of isoprene and basically get what's called a sesquiterpene. Farnesol is an example of a sesquiterpene. Basically, we see three separate or three isoprene units making up this molecule. Or we can bring in even a fourth isoprene unit and make a diterpene, such as phytol. We can make even larger molecules as well. Beta-carotene is an example of a molecule that is a terpene that is actually quite a bit larger than phytol. It actually would be a tetraterpene, which is one of usually the largest terpenes that we see. So some of the important terpenes, squalene is a precursor for cholesterol. It's also found in some types of olive oil, in not high amounts, but in notable amounts. It's not generally enough to cause drastic increases in cholesterol level within the body, but there is a potential that excessive olive oil consumption, I mean very, very high amounts, could potentially be could cause cholesterol. I did mention beta-carotene earlier as being a tetraterpene. Basically, beta-carotene is derived from 8 isoprene units, and essentially it serves as the precursor for vitamin A in our diet. If we look at how vitamin A is produced, basically beta-carotene is oxidized and broken open in the middle of the molecule to yield 2 molecules of retinol that we see right here. Retinol undergoes oxidation from an alcohol to the corresponding aldehyde, which is retinal. Retinal then gets attached to a lysine residue in the molecule opsin to give rise to the molecule that we call rhodopsin. Rhodopsin is the combination of opsin and retinal. And the importance of vitamin A in vision is that retinal undergoes an isomerization between the trans form that we see here and the cis form that we see there. And that isomerization is actually part of the visual process. Basically, it is converted from the cis form to the trans form by light, and then we have this enzyme retinal isomerase that we see right here that puts the trans form back in the cis form. And that continuous recycling is an important aspect of how we perceive light. We can also have mixed terpenoid compounds. These are molecules that contain a portion of the molecule that is derived from isoprene and a portion of the molecule that is not derived from isoprene. So a couple examples of these, vitamin E, which is also known as alpha tocopherol. This portion of the molecule over here is derived from isoprene, whereas this portion right here, which is the actual active portion of the molecule, is not derived from an isoprene. Vitamin E is a membrane antioxidant, so it needs this lipid tail, which is supplied by the terpenoid portion to basically sequester it in the membrane. Vitamin K, which is an important ingredient for proper blood clotting, is also an example of a mixed terpenoid, and basically this portion of the molecule is derived from the terpene portion of the molecule, and the rest of it that actually participates in the reaction, the carboxylation reaction that takes place, actually comes from the non-terpene portion of the molecule. Squalene, which was one of the terpenoids that we looked at earlier, basically gives rise to the molecule cholesterol. Cholesterol has a fused ring structure. As we see here, there's 4 fused rings in the structure of cholesterol, along with this hydroxyl group, which imparts a minimal amount of water solubility to it. Cholesterol is really not soluble in water appreciably at all. However, cholesterol is a precursor for a variety of molecules that actually are water soluble, and the molecules that are derived from cholesterol represent some extremely important molecules within our body, such that cholesterol is something that we really do need in our body. It's just that we don't need the amounts that many people in this country tend to have. So high cholesterol is basically can ultimately be detrimental to our overall health. Some of the important steroids that are derived from cholesterol include things like beta estradiol and testosterone. These are the primary sex hormones, which basically make somebody either male or female. The conversion of testosterone to beta estradiol basically involves just a quick enzyme catalyzed reaction to do that conversion. Males lack the enzyme called aromatase that does this conversion, whereas females do have this enzyme present and can catalyze that reaction. Some other hormones that we see include things like aldosterone. Aldosterone is a mineral corticoid that is responsible largely for regulating the sodium concentrations that we have in our body, although in so doing it also regulates the concentrations of many other ions, such as chloride ions and potassium ions. Cortisol is a glucocorticoid. Cortisol is referred to by many as the stress hormone. We tend to produce a lot of cortisol under very stressful situations. And it basically has an overall goal of regulating our metabolism of nutrients, and possibly, especially under conditions when it's released in response to stress, to try to get our body to prepare for essentially possible times when there might not be nutrients available. Something that's not terribly common now, but something that cortisol's original purpose in evolution was to prepare the body for times when nutrients would not be so available. Nowadays, cortisol probably causes more problems than it helps with humans, simply because of the readily available supply of nutrients that we have in our developed nations. Some other steroids, vitamin D is derived from cholesterol. Basically, This ring here is what would be the A ring. This is the C ring, and this was the D ring from the original cholesterol structure. The B ring is basically broken open to produce this portion of the molecule. The particular molecule we have here is 1,25-dihydroxy vitamin D. It's derived. There are a variety of D vitamins that are created, and I'll actually show you on the next slide kind of the schematic for formation of various vitamin D molecules. A derivative of cholesterol that's actually very important in the absorption of fats and also fat-soluble vitamins from the diet are the bile acids. And basically cholesterol is converted into this molecule cholic acid. And basically what you'll notice is that cholic acid has a couple more hydroxyl groups as well as a carboxylic acid present on this molecule. It's simply to increase the water solubility. And cholic acid acts essentially like a detergent. When we ingest lipids in our diet, we they tend to form fat globules in our intestines, and the bile acids are meant to break up those fat globules so that the fat can be absorbed. The triacylglycerols and phospholipids that are present in the diet can be absorbed and thus utilized as a source of fuel. In so doing, it also breaks up the fat globules that would contain fat-soluble vitamins and allows us to absorb things like vitamin E or beta-carotene from our diet as well. So, although it does, many people deride the bile acids for, allowing us to absorb so much fat from our diet, they do play a very important role in allowing us to absorb fat-soluble vitamins. So vitamin D is a steroid vitamin basically derived from this molecule 7-dehydrocholesterol. And basically ultraviolet radiation causes this bond right here to be broken. And that's the bond that opens up to form the structure that we see here in cholecalciferol, which itself is one of the vitamin Ds. There are several different forms of vitamin D. Basically, cholecalciferol then undergoes an oxidation to form the 25-hydroxy cholecalciferol, which then undergoes another oxidation to form the 1,25-dihydroxy cholecalciferol. All of these have certain levels of activity as vitamin D, although in general this last one, the 1,25-dihydroxy, is really the most active form of vitamin D. What vitamin D does is it generally regulates the levels of calcium and also phosphorus that we have within the body. Largely it's meant to increase synthesis of the calcium binding protein that we find in the intestines. And calcium is something that under normal conditions would not be very well absorbed within our intestine. We actually have a protein that binds to calcium and allows it to be absorbed into the body. A deficiency of vitamin D, particularly in children, can lead to rickets. And rickets is basically without vitamin D, we don't get the calcium levels that we need, and particularly in developing children, we see that the bones do not develop properly. They end up very soft, and as a result, they usually end up with skeletal problems. And it's something that can be prevented fairly easily by either providing vitamin D in the diet or simply having some sun exposure, getting this ultraviolet radiation that's needed. In fact, there's a big debate between a lot of doctors and dermatologists. A lot of doctors claim that dermatologists are going a little bit overboard on the sunscreen issue. There are a lot of diseases that could possibly be prevented by simply having more vitamin D, and getting just a little bit of ultraviolet light exposure on a daily or at least several times a week. Um, so generally about 10 to 20 minutes has been considered by many to not be damaging, and 10 to 20 minutes of ultraviolet radiation, and potentially even very beneficial in terms of the vitamin D that is produced.